Erica runs a lab in the Department of Genetics and is a member of the University of Pennsylvania's Epigenetics Institute. Erica received her bachelor's degree from Yale University in molecular biology, as well as worked as a research assistant at both Yale and the National Institutes of Health, NIH. She traveled across the country and went to the University of California, San Francisco to obtain her PhD in neuroscience, where she worked with Dr. Steve Fink. I apologize if I say it wrong. Fink Biner. I apologize, Steve. Then she traveled back to the East Coast and went to Rockefeller University, where she received her postdoctoral training with world-renowned epigenetic scientist, Dr. David Ellis. She has won several awards over the past years, including the Alfred P. Sloan Fellowship. And outside of the lab, she enjoys playing a mixture of sports, including cycling, ultimate frisbee, and fencing. And I hope today gets a takeaway about how histones, proteins in the brain, are tiny but mighty. Go ahead, Erica.
All right, hello. Just trying to get my video to go back on and it won't enable, so can you guys hear me?
You're good. We can see you and hear you.
Okay, great. Have a little lag. All right. On my screen, I'm not seeing slides come up yet. Can you guys see slides? There it goes. Okay, great. So as you heard, I am going to talk about a protein called histones. And these are some of the tiniest proteins in our cell, but they're also some of the most important. And hopefully by the end of this 15 minutes, you'll know why they're so critical to things like learning. All right, so we work on neurons, as you heard from Vanessa's excellent introduction, and we work on DNA, but not just DNA, but in how it's regulated inside of neurons. So this is a neuron. It's lit up with a green fluorescent protein so that we can see all of these really beautiful structures that the cell makes. And the neuron has in it a big cell body, which you can see here, as well as all these kind of arms coming off of it. And it's these arms coming off where neurons actually communicate with each other and form memories and store information. But inside the cell body, really important things happen as well. And that's what we study. In fact, all of the DNA of the cell resides inside the cell body. So DNA, you've seen in a couple of formats, I'm sure, in pictures where you often see it kind of stretched out in this nice helical structure, but you also see it really tightly packed into these structures called chromosomes, which are maybe what you've seen under a microscope in biology class. But how you get from this strand all the way to this really compact structure involves the proteins that we call histones. So these are these very small proteins that are responsible for wrapping up this incredibly long strand of DNA into these structures so that they'll fit into the cell's nucleus. But also, while they wrap up those DNA strands, they are able to turn on and off genes. So they do this to allow a cell to control how it makes RNA. RNA goes on to make protein, which can then turn into all the things that a cell needs. So my screen just went blank. Can you guys see? Ah, there we go. So this process, going from DNA all the way through to protein, is really the building blocks of a structure, all the structures that make up a neuron. Okay, so I'm going to show you DNA and histones throughout this talk like this, with the histone proteins in gray, wrapping up DNA in black. So DNA kind of gets wrapped around these histones like a thread around a spool. And it's this wrapping and unwrapping of DNA that lets histones regulate genes and turn them on or off just like a cell needs. Okay, so there's a lot of different ways histones can do this. I'm going to tell you about this in terms of ice cream, at the risk of us all really wanting ice cream by the end of this and having ice cream for dinner. So histones start out kind of like these little boring structures you can think of your vanilla ice cream cone right here. Not much is happening. but they can wrap up DNA nonetheless. They can also get sprinkles. So we can put some sprinkles on our ice cream. And this is basically just histones getting these little sprinkles or modifications, these little dots, which I'm showing in different colors here, that let them turn on and off genes just in response to various kind of cellular signals or environmental perturbations or anything that a cell needs to respond to by activating genes. Okay, so you've got ice cream and it can get sprinkles. But there's one other thing a cell can do, and that's what I'm going to talk about today. And it's actually getting a different flavor of histone. So in addition to getting a topping, we can also replace this ice cream with a different flavor. So instead of vanilla here, I'm showing you this kind of very elaborate strawberry rainbow thing going on, where this particular set of histones has swapped out one of its normal histones for a different flavor of histone. And these different flavors we call variants. Okay, so today I'm going to tell you about one variant that turns out to be really important in the brain. So this is a variant called H2B, and it's a variant of a protein called H2B. So there's, if you can catch that, H2B is the normal one, H2BE is the variant. So the normal one, H2B, is the vanilla ice cream here, and it's this top row of amino acids I'm showing you on this line. So the little letters are just different amino acids that encode the protein. And the histone variant, H2BE, is basically made up of most of the same amino acids. So it has, the same kind of cream and sugar and maybe some vanilla in there, and it gets frozen down to make a protein just like your regular H2B, but it also has a little bit of flavor added. So maybe some strawberries get sprinkled in here so you get some nice strawberry ice cream out instead of just vanilla. Okay, so we have two different flavors. And you can see these differences between them. Not a lot of differences, just a couple, and they're sprinkled all over this protein. Okay, so what does this protein do, and why do neurons use it? Well, to answer this question, we began by using mice, which is a system we use in the lab often to study what happens in the brain. And we took some mice and we put them basically in a really fun cage. They got to play with some toys, they got to run around in this box. They had some interesting food here to play with and eat, which they don't normally get, in this case, some banana slices. No ice cream, unfortunately. They have a little pool of water here they can splash around in and drink and so on, and climb on this box or inside of it, and they've got some Legos. So we let them play in this box for a day. And this is a lot more interesting than the normal boring home cage where they usually live. And it activates a whole ton of neurons in the brain. And we looked at what happens to our favorite protein, H2BE, when we put mice in this really fun little space. It turned out H2BE actually decreased. So what you're seeing here is the sort of level of H2BE in normal mice and the level of H2BE in mice that got to go play in this fun environment. And this was pretty surprising. This is not normally what happens in the brain. Usually the brain gets activated by something like this, and it turns on a bunch of things. It activates a bunch of things. It makes more of proteins. But this protein does the opposite. So this got us really excited because something weird is going on with this particular protein. And neurons somehow are regulating it in a very bizarre way. So next we looked at what this histone H2BE does to DNA. So I told you at the beginning that histones, one of their jobs is to wrap up DNA into structures. So to figure out how H2BE wraps up DNA, we looked at some histones that are bound up with DNA that contain either just normal H2BE, so this is histones that just have the vanilla flavor and that's it, or histones that in place of the normal H2B have the H2BE variant. So this is the little strawberry flavored histone stuck in here. And what we found was we looked at these structures using a very special microscopy called atomic force microscopy that lets us look at incredibly small things like these histones. And we see with the regular histone these little bright spots, and this tells us that we're looking at really nicely wrapped up DNA. These histones do a great job of holding up DNA into these tiny little blobs. The histones that have H2BE, or this different flavor, do a lousy job of this. They're spread out. It's sort of like if you dropped your ice cream on the ground on a hot sunny day and it melted everywhere and made a big mess. You can even sometimes see the whole DNA strand unraveled here. So this means that the cell might be using H2BE to unravel its DNA and open it up to allow it to maybe activate genes in certain circumstances. So to figure out what those genes might be, we went on to look at the actual RNA that gets made in neurons when they use these different histones. So I told you at the beginning that we can look at regulation of RNA, and that makes up neurons. And we can also do this by using and compare different conditions in the lab by using what we call sequencing methods. So this just lets us take cells like this one and sequence it to figure out all the different RNA that that cell is making. So we did this with some cells that have all the normal kinds of flavors of histones. They have the regular ones, they have the H2BE, normal neurons that can do whatever they want and whatever they need to. We compared these neurons to neurons that don't have H2BE. So they're missing vanilla, they only have vanilla ice cream, they're missing strawberry ice cream from their repertoire of flavors to choose from. And we measured all the RNA in these cells and looked at the differences. And what we found was that our normal cells had a set, all the normal sets of RNA that encoded the whole protein. When we looked at the cells that had just regular histones and no H2BE, there was a big change in the proteins that encoded these tiny little synaptic structures. So we were able to change something in the nucleus by getting rid of one of these flavors of histones that lives in this nucleus, and that actually affected the genes that would eventually go on to make the proteins that formed these synaptic structures. It was a couple slides ago, so I'll remind you what these do. These structures are where neurons talk to each other and where they store information. So that told us that perhaps this flavor of histone was important in regulating how neurons communicate and maybe even how they store information to allow us to form memories. So the last thing we tested was whether or not this flavor of histone actually had some link to memory formation using mice. So here we did a simple test. We gave mice some time to run around a little box that had two objects, and they got to explore that box and look at these objects and play with them. The next day, we put them back in the same box and gave them one of the objects they'd already seen and a new one. So mice are just like us. They're a little more interested in the new shiny toy they haven't gotten to play with yet. And they'll usually spend a little more time with this new object and a little less time with the old one. But to do that, they actually have to remember what they saw the day before. If they don't remember, they won't know which one's new. And this is what this test actually looks like. Here's a little mouse, and it's investigating this, I think it's a faucet. And today isn't quite so interested in this little Lego that's over here. Maybe it saw this one the day before. So we measure this and look at how mice differ depending on whether or not they have H2BE protein. So this graph right here is showing you a bunch of dots that all are different mice. So these different mice, we measure whether or not they prefer the new object to the old. And if they do prefer it, they wind up on this sort of positive side of the graph. So these are mice that are doing what they're supposed to. And this set of mice right here are normal mice. They have both kinds of ice cream to choose from, the regular and also the histone H2BE variant. This other kind of mice, which we call knockout mice because they're missing a gene, we've knocked out that gene, only have regular ice cream. They don't have any strawberry flavored H2BE ice cream. And they do much worse on this test. They kind of randomly like one or the other, but there's no real preference for that new object compared to the old one. We've done a number of other tests as well that kind of control for some other possible things that could be going on. But what we've learned is that this histone variant seems to be absolutely necessary for mice to be able to form memories. Okay, so now we know that these different flavors of histones wrap up DNA, they control gene expression, and they help an animal learn and store information in their brain. All right, and with that, I'm going to finish, but I really want to show you the most important slide, which is acknowledging all the people that actually did this work. My whole lab really contributed to all sorts of different parts of this project, but in particular, I really want to call out Emily Fearman and Sean Lozon, who've been working on H2BE in my lab and produced much of the data I just showed you here. And with that, I'm happy to take any questions.
Well, thank you, Dr. Korb. That was a great talk. So if you have any questions, again, feel free to put them in the Q&A section, or actually, if you want, you can raise your hand and you can come on stage and ask them in person. All right, so it looks like we have one question here. I'll just announce it just in case anyone can't see the screen, but Ethan Blackwood asks, how do you know that the novel object identification test is affecting memory as opposed to the behavior of looking at novel objects?
That's a great question. So from what I showed you, you wouldn't know that. I just showed you a little part of the data, but there's a lot more data that we measure with these tests. And we look at that. So we want to know whether or not, for example, getting rid of this H2BE just makes the mice not very interested in anything. Maybe they just sit in the corner and don't explore at all, so there's no chance for them to learn. Or maybe it makes them kind of sick and they don't move well, so they can't explore the objects very effectively. So we do a lot of extra testing to see if any of that is happening. In this case, it's not. So we know these mice actually move totally normal. They're very healthy. They explore the objects just as much as the control mice do. So they go up and they interact with all of those objects just as much on the day when they're supposed to be learning, and even the day when we're testing them. It's just that their preference changes. So they no longer have this preference for objects. They don't have a preference for the new versus the old. Suggesting they can't remember. Whereas when we give them 2 objects on day 1, they have a similar preference for exploring those objects as the wild type. So it's a great question, and from what I told you, you wouldn't know, but it turns out it does seem to be pretty specific.
All right, great.
I see another question from Matt. Should I read that out, or do you want to?
You can read that out if you want.
Okay, so Matt asked, do the mutant mice have any other changes in behavior or mood anxiety? So we're doing a lot more testing on these mice now, but I will say so far what we've looked at suggests no. So we've looked at an anxiety measurement by looking at where mice spend time in an open arena. Mice tend to prefer periphery of an arena, but we'll venture into the middle of it, and that sort of preference for periphery versus middle of a space gives us that anxiety readout. They don't show any changes there. We also don't see changes, at least at this age, in a T-MACE test, which is more of a short-term learning test. So their short-term learning seems to be intact. It's really this long-term memory deficit. All right, I see another couple coming up. So what is the difference between H2B and H2BE? So the differences are pretty subtle. There's just 5 amino acids that differ, and the whole protein is maybe 120 something amino acids. So those aren't big differences at all. Those are kind of tiny as far as different proteins go. So you might expect it actually not to matter at all, but it turns out those specific amino acids land in some pretty important places in the protein. So they land in some of the places where the histone actually is linked up to DNA. So we think that perhaps some of those amino acid changes cause the chromatin, the histones, to bind differently to DNA, and maybe that's how it opens up that DNA structure. They also actually can have sort of different types of sprinkles. I didn't get into that, but the regular histone, the sort of vanilla flavored histone, can have a certain type of sprinkle, and this particular modification called ubiquitination. And it turns out that the H2BE. All right. I also see a question about have we found any differences in age, young versus old, from Vanessa, and that's another great question, and gives me an excuse to tell you a little bit about what Sean's been working on in my lab. So I told you a lot about how H2BE works in neurons, but we've also looked at how it works in another cell type in the brain called glial cells, particularly in aging. And it turns out in those cells, these are kind of support cells in the brain, but they do really important things and help control neurons and their function. And they're also often linked to aging-related disorders, at least in some contexts. It turns out in those cells, H2BE seems to control the ability of glial cells to divide. And what that ultimately means in the aging context is that getting rid of H2BE actually can help cognitive function late in age. So if we look at old mice, we actually see very different things. I told you about young mice today, but we change the age, the behavior is quite different. I don't want to go too far over my time, but should I take another question or two?
I think we have time for another question or two.
Okay, great. So the next question is from Andrew about have we investigated differences in H2BE mice with abnormal memory conditions, such as amnesia, compared to normal mice? If not, do you think these histones levels may have any influence? So we haven't looked at this exactly. So the idea would be to look at, say, a neurological disorder of some sort, I think, in mouse models of some of those disorders, and see what happens with H2BE. But again, Sean in my lab is actually trying some of this by looking at some basically Alzheimer's related mouse models and seeing how H2BE may be affected in those neurological disorders. And then next question I'm seeing here, oops, they keep moving on me. Do we notice if isoforms are subject to different modifications other than ubiquitin from Georgia? So we've primarily focused on H2B ubiquitination. That's one of the most well-studied modifications on H2B. And in that case, it turns out H2BE, the variant, actually cannot get ubiquitinated, whereas the normal protein can. We haven't seen major changes other than that, but we also haven't looked very closely. And there's not actually a lot of histone modifications on H2B that are that well studied. So that's the sort of big one that everyone looks at on H2B. And there's not a lot of other ones with a ton of data out there, or more importantly, with a ton of really good reagents. So we're kind of reliant with these studies on the tools that let us detect them. And so, for example, the ubiquitin site has really great antibodies, and it lets us study that very carefully, whereas some of the other modifications a little harder. But we do want to certainly explore more and see if there's other modifications that maybe only show up on H2B.
Okay. Thank you all for the really great questions. So now we're going to move on to our next speaker, but for those whose questions didn't get asked, you can stick around, or didn't get answered, you can stick around until the end of the 3 talks, and we'll have a Q&A panel where we can revisit some of these questions. So again, thank you so much, Dr. Korb. And now we will be going on to our next speaker.